Hello students, let's continue the topic. Now, germination. We know that seed contain embryo, and this embryo is present in dormant stage. Dormant stage means inactive state. If we see the seed from outside, it appears that it is without life, but even the dry seeds, they also take oxygen and give out carbon dioxide. It means the chemical reactions are still going on in the dry seeds also. Whenever these seeds get favorable conditions, they germinate and give rise to a new plant. Now we will study the conditions necessary for germination. The first condition that is necessary for germination is the presence of water. Water is required because when the seeds absorb water, they swell up and their outermost covering ruptures so that the radicle can come out to give rise to a root system. Second importance is that inside the seeds, the food is stored either in the form of cotyledons or in endosperms. Now, to make it in diffusible form, water is required. When it comes in diffusible form, then only the embryo can utilize that food. So, to make it in a diffusible form, water is required. So, water is required for two important things. Number one, it is required so that the testa can rupture and the radicle can come out. And second is, it converts the stored food into diffusible form that can be easily utilized by embryo. Second is suitable temperature. At very low and very high temperature, seeds do not germinate. Seeds germinate generally at the moderate temperature that ranges from 25 degrees to 35 degrees Celsius. Because at very high temperature, the tissues, the delicate tissues, they are destroyed, and at very low temperature, the growth of the embryo is inhibited. So the suitable temperature is required for the germination of seeds. Third is the presence of oxygen. During germination of seed, the cell division and the cell growth takes place. And we know that both of these processes require energy. And from where we get energy? We get energy by the process of respiration. And what is required for respiration? Oxygen. So this oxygen is required for respiration to get energy that can be utilized for cell division and cell growth. Now here are some experiments given related to germination of seeds. First experiment is to prove that water is necessary for germination. It's a very easy experiment. You can do it at your home also. Simply, you have to take two beakers, place wet cotton at the bottom of both the beakers. Place wet cotton at the bottom of beaker A, and at the bottom of beaker B, place dry cotton. Now place some seeds over these cotton present in the beakers. Observe them after few days. You will see that in beaker A, the seeds will germinate, while in beaker B, seeds will not germinate. This shows that water is necessary for germination of seeds. Second experiment is experiment to prove that a suitable temperature is necessary for germination. Now again, do same. Take two beakers. Now place wet cotton at the bottom of both beakers A and B. Now place some seeds over the cotton. Now place one beaker at room temperature and other beaker inside the refrigerator, and observe after some days. You will see the beaker placed at room temperature will show germination of seeds, while the beaker that you have kept in refrigerator, the seeds will not germinate in it. So this proves that a suitable temperature is required for the germination of seeds. Now, third experiment is to prove that air is essential for the germination of seed. This is the setup shown in the diagram. You have to take two conical flasks, A and B. Place wet cotton at the bottom of both of these. Now, in beaker A, place some seeds here and hang one little test tube having plain water in it, and then place a cork over the mouth of this conical flask. In beaker B, place wet cotton, put some seeds. Now, instead of putting plain water in this small test tube, you have to put alkaline pyrogallic acid here because it absorbs the oxygen. So, whatever the oxygen will be present here, it will absorb it. So, seeds will not get oxygen. Okay. Now, again, place a cork here. Now, observe this experimental setup. After a few days, you will see that the seeds will germinate in conical flask A, and they will not germinate in conical flask B. This indicates that oxygen is required for germination. These seeds will not get oxygen because all the oxygen was absorbed by pyrogallic acid. That's why they will not get oxygen, while here they will get oxygen, so they will germinate. Now, one more experiment is there. This is called 3-bean experiment. You have to put the bean seeds, place the bean seeds over a wooden plank like this and put it inside the water. Now, this seed is getting water, but it is not getting oxygen. This seed is getting water as well as oxygen. This seed is getting only oxygen but not water. So, when you will observe after a few days, you will see that this middle one will germinate only because it is getting both water and oxygen. This seed is not getting oxygen, so it will not germinate. This seed is not getting water, so it will not germinate. So this is called 3-seed experiment.